Good evening. I am Dr. S. Sudhagar, Biology faculty from SURIYA Academy, Salem. Before starting my new chapter, I will give some tips to crack Neet. Short-term study, somewhat difficult to get more marks, but which give experience to study. First, we must study NCERT 11th to 12th, I mean +1, +2 books thoroughly. It helps to get high marks, but coaching center books also supports to get 360 marks in biology. Regular coaching classes promotes to study shortcuts to easy romper. I mean, what I am trying to say means, when you are going to join in coaching classes, which promotes your study in the study interest level, we must study easiest topic first, which also enhance the interest of study. We must study all chapters. Then we must prepare NCERT based questions. And we must prepare question bank and MCQ based on the NCERT syllabus, which enhances our score up to 360 in biology. That NCERT based questions, question banks and MCQs will discuss in our year-long program, because the time is short. Now we cannot discuss due to the short time. Okay, let's discuss our today's topic. Today we are going to discuss about the principles of inheritance and variations. What chapter we are going to discuss? Inheritance and variations. So you know very well, in biology is very easy compared to the other subject. However, so if you see the literal meaning, easily find the answer in biology. When you are seeing the literal means, easy to find the answer. For example, biology. Bio means a living. Logy means a study of living things, like that. Study of living things, which is known as a biology. Like that, we must see the literal meaning. So when you are going to study a single chapter, first of all, you should know what are the things which is present in the
Sir?
Hello.
Okay, sir. Continue பண்ணுங்க sir.
Sir, இப்ப கேக்குதுங்களா?
Voice okay sir, clear.
Okay. Okay. Sorry.
Okay.
Okay, today we are going to discuss about the inheritance and variations. So inheritance means, just now I told, pathway of genetic traits and expression from one to another generation. So traits means specific character of individual. Traits means nothing, specific character of an individuals. So individual having a specific character, that is called as a traits. That terminology is a very, very important. So inheritance means a pathway of traits which is transmitted from the one to next generation. That character also should be expressed in the one generation. That character also should be expressed in the another generation. That is called as a inheritance. Clear? So again, I repeat the definition. Pathway of genetic traits and expression from one to another generation is known as a inheritance. Clear? The next one is a variation. So what's the meaning of variation? So variation means nothing. It is a changes or differences. That is called as a variation. That variations, the differences occurs in the same species. The differences occurs in the same species or differences between the individuals or group of organism, which is known as a variation. Clear? So variation in the same species or variations, differences between the individuals or group of organism is known as a variation. Clear? That variations caused by the majorly two factors. What are they means? One is a genes, another one is a environmental factor. One is a genes, another one is a environmental factors. In case variation is caused by the genes, that is known as a genotypic variation. In case variation is caused by the genes, which is known as a genotypic variation. If the variation is caused by environmental factor, in case variation is caused by the environmental factor, which is known as a phenotypic variation. Clear? Which is known as a phenotypic variations. So before entering that topic, you should know some important terminology. I will give the outline only. Remaining things we will discuss in detail. So first one is a factor, allele, homozygous, heterozygous, phenotypic, genotypic, back cross, test cross. Those are the important terminology. We are going to discuss it. Clear? So that inheritance and variations, that topics comes under the genetics. That topics comes under the genetics. So genetics, what's the meaning of genetics? So just now I told we should see the literal meaning. We should see the literal meaning. So genetics, we have split the word. We can split the word genetics. Gene. What's the meaning of gene? What's the meaning of gene? Functional part of chromosome. Gene means nothing. It is a functional part of chromosome. Is known as a gene. That gene only determine the character. Gene only determine the character. The hereditary units also called as a genes. Again, I repeating functional part of chromosome. Hereditary units also known as a genes. Clear? So in genetics, we are going to discuss about the genes function, how chemical composition, etc. Those things we are going to discuss in the genetics. Clear? So genetics, simply I will give one definition for genetic. The scientific study of genes and heredity is called genetics. The scientific study of genes and heredity is called as a genetics. What is the meaning of heredity? Just now I gave one definition for inheritance. Now I am going to give definition for the heredity. What's the meaning of heredity? Transfer of character from one to another generation. So transfer of character from one generation to next generation, which is known as a heredity. Clear? That particular character will be transferring from one generation to next generation. That is called as a heredity. Inheritance means a pathway of genetic character. Clear? That is the major difference between the heredity and inheritance. Clear? That is the, you should understand the difference between the two. And W. Batson in the year of 1905. This one, ma. Then green color, which is noted. William Batson, W. Batson, in the year of 1905. He only coined the term genetics. Same time, he only very first used the term genetics. So in case that question may ask like this, who very first used the term genetics? Answer is W. Batson. Otherwise, the question may ask like this, who coined the term genetics? Answer is the same, W. Batson. So the term genetics coined and used first time by the W. Batson in the year of 1905. That is also important Neet question. Then who is father of genetics? Everybody know who is father of genetics? Gregor Johann Mendel. Gregor Johann Mendel. In red color, which is denoted. Gregor Johann Mendel is called as a father of genetics. Why he is called as a father of genetics? Why he is called father of genetics? Very first, now we know very well DNA is responsible for all, but olden days, we don't know. Olden days, we don't know. So very first, DNA is responsible for all. The character will be transmitted from the gene. I mean DNA. The character will be transmitted from the gene of DNA. The character will be transmitted from the one to next generation through the gene. That thing is very first explained by the Gregor Johann Mendel. That's why he is called as a father of genetics. So that question also important questions. Who is father of genetics? Who coined the term genetic? That is a important questions. Next one, selective breeding and artificial breeding helps to develop the desirable characters. Selective breeding. You know very well, breeding means to produce the more things by crossing or any other things. Okay, so selecting breeding or artificial breedings helps to develop desirable characters in plants or maybe animals. So by using the artificial breeding, Shahiwal, by using the artificial breeding, by using the artificial breeding, Shahiwal. Shahiwal means nothing. It is a cow, Shahiwal cow, which is produced from the wild variety of cow. Wild variety of cow. What purpose Shahiwal is produced? What purpose Shahiwal cow is produced? Means to get a high yield of milk, high quantity of milk. That is also important question. Shahiwal cows produced from the wild variety of cow for the production of a high quantity of milk. The next one, we must know about Gregor Johann Mendel. Few important things only, which is related to the Neet. Mendel, Gregor Johann Mendel. He was born in the year of 1822. He was born in the year of 1822 in Austrian. So contribution of Mendel to the genetics, which is known as a Mendelism. Contribution of Mendel to the genetics is known as a Mendelism. So Mendel, he did so many experiments. Based on his experiment, so many things will be explained. So that is called as a contribution. That is called as a contribution. The contribution of Mendel to the genetics, which is known as a Mendelism. That is also important question. So Mendelism means contribution of Mendel to the genetics, which is known as a Mendelism. So Mendel took Pisum sativum plant. Pisum sativum, that is a botanical term for a garden pea plant. That is a botanical term for garden pea plant. He took Pisum sativum plant for his experiment. That is also important question, Pisum sativum. He took Pisum sativum plant. Common name is a garden pea plant for his experiment. The Mendel's experiment, everything published in one book named as Experiment on Plant Hybridization. So Mendel's work published in Experiments on Plant Hybridization. in the year of 1866. In the year of 1866. So Mendel, he selected 22 varieties of cotton pea plant. Mendel, he selected 22 varieties of cotton pea plants. Kindly take a hints, ma. So during the exam time, that hints will be support to you. So kindly take a hints. So Mendel selected 22 varieties of cotton pea plants. That is also important question, ma. 22 varieties he selected, and he selected 7 characters. He selected 7 characters, 22 varieties of cotton pea plant. He did his experiment for 7 years. He did his experiment for 7 years, up to 1856 to 1863. He did his experiment for 7 years, 1856 to 1863. That Mendelian principles and discoveries were rediscovered by 3 botanists. The principles of genetics was rediscovered. So the particular thing that monohybrid, dihybrid, a lot of experiment did by the Mendel. Lot of experiment did by the Mendel by using the pea plant. And finally, he explained the gene is responsible for everything. He find it. So after that, contributions, Mendel contributions, Mendel's discoveries, which is rediscovered by the 3 botanists, namely Cornish and Deveries and Diskermark. That 3 botanists are rediscovered. That is also important question, ma. Again, I repeat, principles of genetics was rediscovered by 3 botanists, namely Corins, Deveries and Diskermark. Those are the importance of Krieger Johann Mendel. The next subtopic is a uniqueness of Mendel experiment. Next subtopic is a uniqueness of Mendel experiment. Uniqueness of Mendel experiment. Mendel experiments involved in 4 steps. So majorly Mendel experiments involved in 4 steps. What are they? One is a selection. Second one is a hybridization. Third one is a selfing. Fourth one is a calculation. So Mendel experiment involved 4 steps. First one is a selection. So what is the meaning of selection? Selection means he selected the Pisum sativum plant. Why he is selecting the Pisum sativum plant? Lot of plants are available. So among that many plants, why he is selecting the Pisum sativum plant? Because so many reasons. Few important reasons are, pea plants are easy to cultivate it. Pisum sativum plants are easy to cultivate it. The Pisum sativum plants are self-fertilization. The self-fertilization, I mean, self-pollination will be occurs easily. Self-pollination will be occurs easily. Similarly, artificial fertilization also successful. Artificial fertilization also successful in Pisum sativum plant. Similarly, that pea plants growth period is too short. Pisum sativum plants are short growth period. Those are the important reasons for selecting the pea plant for his experiment. This is called as a selection. So first steps, selection means nothing, selecting a plant, experimental plant. Second one is a hybridization. Hybridization means, you know very well, 2 different things will be crossing or combined together. That is called as a hybridization. For example, this is Next one is a uniqueness of Mantel's experiment. Next one is the uniqueness of Mantel experiment. So uniqueness means individuality. That is called as a uniqueness. Significance. We can say in other words significance. So uniqueness of Mantel's experiment. So Mantel's experiments involved in the 4 steps. Just now I explained selection. Selection means selecting the plants. The Pisum sativum plant we selected for the so many reasons. Just now I explained. The next one is a hybridization. Hybridization. What is the meaning of hybrids? Hybrids. Hybrids. Hybrids means nothing. The 2 different things will be combined together. 2 different things will be combined together, which is known as a hybrids. So hybridization means 2 things will be combined. That is called as a hybridization. For example, tall plant and a trough plant. Tall plant and a trough plant. 2 characters will be combined together. That is called as a hybridization. That hybridization also the second step. The next one is a selfing. Selfing means you know very well that for example in monohybrid cross in F1 generation we can get a heterozygous. So like this. So we can get a heterozygous caps T and a small t. We can get a heterozygous caps T and a small t. So that caps T and a small t is undergoes a selfing process, means a self-fertilization process. So at that time the crossing will be again takes place. That is called as a selfing. That is called as a selfing. That is a third point. The last one is a calculation. That last step is a calculation. Calculation means we know very well that we are calculating the ratio, phenotypic ratio and genotypic ratio. That is called as a calculating. So 4 steps are important steps in Mendel experiment. First one is a selection, second one is a hybridization, third one is a selfing, fourth one is a calculation. Next, Mendel, apart from that 4 steps, what are the other uniqueness means? Mendel selected only 1 character at a time. Mendel, he selected only 1 character at a time. For example, if he selected many characters at a time, means the result may be good or bad. We don't know very well, but he selected only 1 character. That is another reason for his success of his experiment. Clear? He maintained a statistical record of the results. Mendel, he did so many experiments that experiment results are maintained in a statistical way. He maintained a statistical way. That is also another reason for reasons for his success. He used a large sampling size. Which gave greater creativity to his result. He done experiments on successive generation. He chose 2 opposite traits. 2 opposite characters. What are they? For example, height. Height means tall and short. Tall and rough. Like that. That is a 2 opposite. Similarly, flower color means violet or white. That is called as the 2 opposite traits. Clear. The pod shape means a wrinkled or a flat-ended, like that. That is called as a two opposite traits. Mendel selected a 14. Mendel selected a 14 true breeding pure line plants. He selected 14 true breeding pure line plants. Those are the some important uniqueness of a Mendel's experiment. The next subtopic is a character selected by Mendel. Character selected by Mendel. So remaining things already explained. He selected the four. Totally, he selected 7 pairs of characters. The book names also already explained. So next subtopic is what are the characters he selected? He selected only 7 characters for his experiment. That is also another reason for his success. He selected only 7 characters. So what are the characters means? First one is height of plant. First one is height of plant. That is a character that each character has 2 varieties. Each character is having the 2 varieties. That varieties which is also known as alternatives. Each character having the 2 alternatives or varieties. For example, plant height means tall and rough, like that. That is called as a alternative. The 2 alternatives are one is a dominant, another one is a variant. Again, I repeat, so he selected only the 7 characters. The characters has 2 alternatives. The character has 2 alternatives. The 2 alternatives out of 2 alternatives, one is a dominant, another one is a recessive one. Clear. So, for example, what are the characters means? The first one is a plant height. Plant height, 2 alternatives. What are they? Tall and rough. Tall is a dominant one. Rough is a recessive one. The next one is a flower color. Flower color, 2 alternatives is a violet. Violet is a dominant one, white is a recessive one. Flower position, the third character is a floral position. Position means where the flower is located, that is called as a floral position. One is axial, another one is a terminal. Terminal means end. So axial is a dominant character, terminal is a recessive characters. Next one is a pod color. What's the meaning of pod? P-O-D. Pod means nothing. The seed closed with a seed coat. That is called as a pod. The seed is closed with a seed coat. That is called as a pod. The pod color is a 2 alternatives are a green and another one is a yellow. The green is a dominant character. Yellow is a recessive characters. The next one is a pod shape. Pod shape. So pod shape is a inflated. I means like a smooth one, inflated. Another one is a constricted. Another one is a constricted. Constricted means somewhat zigzag. That is called as a constricted. Opposite to smoothness, inflated. That is called as a constricted. Then next 6th character is a seed color. Seed color is a yellow. Another one is a green. Yellow is a dominant character. Green is a recessive characters. Next one is a seed shape. Seed shape. Shape means that shape is round and another one is wrinkled. Round is a dominant character. Wrinkled is a recessive characters. Those are the 7 characters he selected for his experiment. Next one is a monohybrid cross. Monohybrid cross. So that monohybrid experiment. What's the meaning of monohybrid experiment? Means crossing of 2 plants. Crossing of 2 plants differing in 1 character is called monohybrid. What's the meaning of mono? You can split the term mono. Mono means a single. Hybrid means just now I told. Hybrid means combining 2 different things. Combining 2 different things. What are the 2 different things we are going to combine it? One is the tall, another one is a rough. Tall is a 1 character. Rough, short is another 1 character. That is called as a hybrid. We are going to mixing the tall and short. That is called as a hybrid. Mono means a single. So monohybrid cross means the crossing of 2 plants differing in 1 character. What is the 1 character? Mono. 1 character means a height. Height is a single character. That alternate is only tall and short. I think clear. Okay. So crossing of 2 plants differing in 1 character is called as a monohybrid cross. It's known as a monohybrid cross. He selected one character height. One is a tall stem, another one is a trough. Trough means a short, SHORT, short stem. So which is considered as a parental one? The tall stem and the trough stem, which is considered as a parental one. The parental one is denoted by the caps letter P. That gametes, I mean factors, are a caps T and a caps T, which is responsible for the tall plant production. Another one factor, small t and small t. Another one factor, small t and small t, which is responsible for producing the trough plants, which is responsible for the trough plant. That we know very well, that which is undergoes the meiosis stage. Meiosis means a reduction. Reduction means that half, that quantity will be reduced in a half. That is called as a meiosis. Clear? The which is undergoes the meiosis stage. While the when that will be occurs, when the gametes will be formed, at the time meiosis will be takes place. So at that time that factor caps T, so will be reduced. So small caps T, small t, which is responsible for the trough, that small t will be which is undergoes the reduction stage, small t. So F1 generation caps T is crossed with the small t. Finally we can get a caps T and small t. This one caps T and small t. The caps T and small t. So in F1 generation we can get a tall plant only. F1 generation we can get a tall plant only. That the second one. So just now I told first after selection, 4 steps are important in Mendel experiment. The second one is a hybridization. So hybridization is over. The third one is a selfing. So selfing means in F1 generation, I mean the self-pollination, that is called as a selfing. The caps T and small t, that is a F1 generation result. That caps T, small t will be crossed with the selfing, self-pollination, caps T and small t. At that time, it is a heterozygous. Heterozygous means, what's the meaning of heterozygous? Hetero means a different. Hetero means a different. Zygous means a zygote, gamete, okay? So different gametes are present, different zygotes are present. That is called as a heterozygous. So heterozygous means a caps T will be separated, small t will be separated. Similarly, here also caps T will be separated and small t will be separated. So we are getting the 4 gametes. We are getting the 4 gametes in the F2 generation. The F2 generation, what will happen? So again, that male crossed with a female one. So caps T will be crossed with a female one. So we can get it a caps T and caps T. And small t will be crossed with the caps T. So we can get it a caps T and small t. And again, this one crossed with the small t, crossed with the caps T, we can get it a caps T and small t. Small t crossed with the small t, we can get it a small t and small t. So end of the F2 generation, we can get it a caps T, caps T, caps T, small t, caps T, small t, small t and small t. You can visualize in the slide, can visualize in the slide. Clear? So at the end of the result, we can get it a 3 tall plants. First one, second one, third one. So last small t, which denotes the trough plant. So phenotypic ratio is a 3:1. 3:1. What's the meaning of phenotypic? What's the meaning of phenotype? So phenotype means morphological characters. Morphological means visualize. For example, what boys look like a black or color or a height, tall or trough, like that. So we can, if you are visualizing, we can easy to say. So that is called as a visible characters. Simply we can say visible characters are called as a phenotypic characters. So while we are visualizing, at that time 3 plants are tall, 1 plants are a trough. 3 plants are a tall, 1 plant is a trough. That is called as a phenotypic ratio. So what are the, how many plants are tall? First one, second one, third one. Because caps T will be present. That is called as a tall. The next one is a small t. It's a 1. That's why the phenotypic ratio is a 3:1. 3:1. That is a phenotypic ratio. The next one is a genotypic ratio. So phenotypic ratio means simply we can say visible character morphology. That is called as a phenotypic ratio. The next one is a genotypic ratio. Genotypic ratio means, so we must see the gametes. gametes. Phenotypic means only we can visualize the outer appearance, but the genotypic ratio means we have to consider the gametes, gene level gametes. The first one, CAMST, that is called as a purity. In genotypic ratio, simply we can say we must consider the purity. Purity. What's the meaning of impure? If you're taking a water, pure water means does not consist of anything. That is called as a pure. That is a pure water. Impure means that water may consist of any other thing. Addition of some foreign particles. That is called as a impure. Like that, purity means the first one, CAPST. Only which having the pure. CAPST only. Clear? That's why we wrote only one. Second one, CAPST small t. There is a heterozygous one. It is not a pure. It is not a pure. It is a impure. Impure means we are mixing of two different things. CAPST and another one is a small t. Two different gametes are mixed together. So that how many things are mixed together like this? One is a CAPST small t, another one is a CAPST small t. So totally two gametes are heterozygous. That's why we are putting a 2. The next one is this is also purity. Small t. So whatever may be, it is a CAPST or it is a dominant or recessive one. So it should be pure line. So we can consider this is a small t is a purity. Small t is a purity. So the ratio is 1:2:1. So the genotypic ratio is 1:2:1. No need to memorize, we can easily understand. So phenotypic ratio means visible characters. Genotypic ratio, the plants are look like a tall only. When you are separating the gametes and visualizing, at that time it is a heterozygous only. So we can say the genotypic ratio is 1:2:1. That is an important question. In monohybrid, phenotypic ratio is 3:1. In monohybrid, genotypic ratio is 1:2:1. That is a genotypic ratio of monohybrid cross. A heterozygous tall. How many homozygous? What's the meaning of homozygous? Yes, now I told homo. Homo means a same. Homo means a same. Zygous means a zygote. Zygous means a zygote. So homozygous. Similarly, that gametes are the same gametes will be repeating. That is called as a homozygous. Homo means the same. So how many things will be present? Only one. So heterozygous trough. Homozygous trough. How many homozygous troughs? Only one. The last one. Small t. So TT. Heterozygous tall. Heterozygous means that is a tall plant, but gametes are a different one. That is called as a heterozygous. TT. So CAPST small t. Totally 2. Clear. So at the end of the monohybrid result in person, ratio just now I told phenotypic ratio 3:1, genotypic ratio 1:2:1. But in percentage, we can say totally 75% of tall plant. We can get a 75% of tall plant. We can get it. 25% of trough plant. We can get it. 25% of trough plant. We can get it. At the end of the monohybrid cross. So among that 75%, 50% among the 75%, 50% of tall plants are heterozygous. Remaining 20% among 50% of tall plants are zygous. Remaining 25% tall plant is a homozygous one. At last, I explained. The last 25% is a so this is a ratio. So the ratio in percentage, we can say 75% of tall plants, 25% of trough plant, we can get it. The next one is in that thing will be expressed in the Punnett square. Punnett square. Suppose Punnett square means the gamete production and zygote formation is explained by the diagram. The gamete production and zygote formation is explained in the diagram. Is called Punnett square, which is also known as a checkerboard. Why which is called as a Punnett square means why which is called a Punnett square means the diagram, the checkerboard is designed by the Reginald C. Punnett. Reginald C. Punnett. That is also important question. The Reginald C. Punnett. He only designed the checkerboard, the diagram method. That's why which is called as a Punnett square. The man-out hybrid cross will be explained in the checkerboard method. Caps T small t, there is a parental one. It will be crossed at the time. We can get it a caps T small t. Caps T, that ratio is a 3:1, 1:2:1. That is a genotypic ratio. Just now I explained. What are the things explained by me? That one is explained in the same thing will be explained in the Punnett square. That's all. The next one is a test cross. Next one is a test cross. What's the meaning of test cross? Test. What's the meaning of test? F1 generation is crossed with the recessive parents. F1 generation is crossed with the recessive parents. So in monohybrid cross, F1 generation. So F1 generation, what we get? Caps T and small t. Caps T and small t. We got only caps T and small t. This one. Caps T and small t. That is a F1 generation result. That F1 generation result is crossed with the recessive parent. Recessive parent. Recessive means a small t and small t. That is called as a recessive. So you should know the terminology dominant and recessive. What's the meaning of dominant? So dominant means which character is expressed. That is called as a dominant one. Which character is expressed, which is known as a dominant one. Opposite to dominant, that is called as a recessive. Which is not exposed, that is called as a recessive. Which is not expressed or exposed, that is called as a recessive. In monohybrid cross, trough, short plant, that is considered as a recessive one. So in test cross, F1 generation crossed with the recessive parent. Recessive parent. This cross is used to test heterozygosity. Test cross is used to test heterozygosity of F1 plants. Hence, which is named as a test cross. Again, I repeat, test cross is used to test. Test means check heterozygosity of a F1 plants. Hence, which is known as a test cross. So we are going to check the F1 plants heterozygosity. That is called as a test cross. Another important point is we are crossing the F1 monohybrid F1 result, caps T and small t, which is crossed with the recessive parental one. I mean small t and small t. It is called as a test cross. So you know very well that gamete will be separated. Gamete will be separated. Caps T and small t. That factors will be separated while new gamete will be formation. At that time, before the gamete formation, what will happen? That gamete will be separated. Caps T will be separated and small t will be separated. Small t will be separated. And that small t also separated like this. So it will be crossed with each other. Male crossed with the female one. So caps T, small t crossed with the caps T, small t crossed with the small t, like this. Clear? So at the end of that result, we can get it a 1:1 ratio. 1:1 ratio. Caps T, small t, and small t and small t. So that end of the test cross ratio is 50% of plants are tall, remaining 50% plants are trough plant. So ratio is 1:1. That is called a test cross. Next one is a back cross. So back cross means crossing of F1 hybrid with any one of its parents. Listen, ma. Crossing of F1 hybrid. F1 hybrid means you know very well that same thing. caps T and small t, that is called as F1 hybrid. Crossing of F1 hybrid with any one of its parent. Any one of its parents means we know very well in monohybrid parent means only two. One is a caps T small t. One is a caps T small t is a dominant parent. Another one is a small t is a recessive parent. Only two parents we are using in a monohybrid cross. Clear. So back cross means nothing. Crossing of F1 hybrid with any one of its parents. So that is called as a back cross. So that back cross is classified into the two types. The back cross are two types. What are they means? Dominant back cross. Another one is a recessive back cross. One is a dominant back cross. Another one is a recessive back cross. So dominant means simply you can say the answer. Dominant means that F1 generation. That F1 generation is crossed with dominant back cross. F1 generation is crossed with the dominant parents. F1 generation is crossed with the dominant parents, which is called as a dominant back cross. Clear. So at that time we can get it like this. So caps T small t is crossed with caps T small t is crossed with caps T small t is crossed with the caps T. So that gamete will be secreted. So we can get it end of the result. Caps T is one gamete. Another one is caps T small t is another zygote. Listen ma. So dominant back cross end of the result is two zygote will be formed. One zygote is caps T. Another one zygote is a caps T small t. Clear. So 100% of dominant character will be exposed in a dominant back cross. So 100% dominant character will be expressed in the dominant back cross. In the recessive back cross, just now I explained in a test cross. 1:1 ratio. 50% of tall, remaining 50% is a trough one. Clear. So I think now you are clear about it, about back cross and test cross. Clear. So test cross means F1 will be crossed with only the recessive characters. Back cross means F1 result will be crossed with either dominant or recessive. If dominant crossed with the dominant means dominant back cross. If crossed with recessive means a recessive back cross. Clear. That's all about the back cross and test cross. That's all about back cross and test cross. Next one is a Mendel's law. Mendel's law. Mendel's law. What's the meaning of Mendel's law? So based on his Mendelian monohybrid and dihybrid experiment, he formulated three laws. So based on Mendelian monohybrid and dihybrid experiment, he formulated three laws, namely law of dominance. First law. Second law is a law of segregation. Third law is a law of independent assortment. So based on the Mendel experiment, the law will be three types. What are they? Means a law of dominance, law of segregation. Third one is a law of independent assortment. The first we will discuss about the first two laws. Why means the first two law is formulated based on the monohybrid cross. The first two law is based on the formulated based on the monohybrid cross. The first law, law of dominance. First law, law of dominance means each organism is formed of bundle of characters. Each organism is formed of bundle of characters. Each and every organism is formulated bundle of characters. Each character is controlled by the pair of individual units. Listen, ma. Each organism is formed of bundle of characters. Each character is controlled by pair of individual units is called factors. So what's the meaning of factors? What's the meaning of factors? Each and every character is controlled by the pair of individuals. For example, caps T, that is called as a factor. Caps T, that is called as a factors. Caps T responsible for what? To produce the tall plant. So that caps T, that is called as a zygote, which is known as a factors. That factors also known as the genes. That factors also known as the genes. That factors occurs in pairs. That factors occurs in pairs. One is dominant, another is recessive one. That factors occurs in the recessive one. For example, here that is by the pair of individual units. The caps T is one unit, another one is a small t is another one is. The pair of character will be controlled by the pair of individual units. Okay, that factors are known as genes. Here, caps T and small t. Which one is the dominant one? We know very well. Caps T, tall plant, that is called as a dominant one. Small t is a recessive one. So one factor in pair, one factor will be prevent or mask the another factor, which is called as a law of dominance. So here caps T and small t, that in zygote caps T and small t will be present. Any one character will be expressed, either caps T or either small t will be expressed. So here the caps T will be expressed. Small t is not expressed, which is known as a recessive one. Which character is not expressed, which is known as a recessive one. So what will happen there? So one character will be expressed. So another character is not expressed. Means what? What will happen there? So one gene is suppressed the another gene expression. One gene is suppressed the another gene expression, which is called as a law of dominance. Again, I repeat more. One in pair, one factor may prevent the expression of another factor. One factor prevent the expression of another factor, which is known as a law of dominance. The particular things will be expressed. That is called as a law of dominance. The next one is a law of segregation. Next one is a law of segregation. Law of segregation. So law of segregation means this law also formulated based on the monohybrid cross. So just now I told the first two laws, law of dominance and the law of segregation. Both law is formulated based on the monohybrid cross. Here in law of segregation, alleles do not show any blending. Blending means joining. Alleles does not show any joining. During the gamete formation, two factors are character. During the gamete formation, two factors of character separate. And enter into the another character. This law is called as a law of segregation. That law of segregation also called as a law of purity of gametes. Listen, ma. Here there is a red color. This is one factor. Here during that zygote formation, here gametes are separated. Here also gametes are separated. Similarly, here also you can see, here this is a heterozygote. This is a heterozygote factor. Caps R is responsible for the red color flower. Small r is responsible for the white color flower. So here caps R will be separated. Small r will be separated. This process is called as a law of segregation. Segregation means a separation. So for what purpose it will be separated? To form a new gamete. To form a new gamete. So that purpose only it will be separated. That purpose only it will be separated. That thing only just now I explained. So that gametes will be separated. That gamete will be separated. After that, it will be ended and form a new zygote. Here, purity. Why it is called as a law? Why which is called as a law of purity means? Why which is called as a law of purity means? Just a minute. Here, this is the heterozygous ratio. Heterozygous means that is a impure one. Heterozygous means that is a impure one. Impure means two different gametes will be present. That is called as a impure one. But while it is a secreted, so at that time, the each gamete can show the individuality only. That's why which is called as the law of purity. Law of purity. So law of separation also called as the law of purity. That's all about the law of segregation. The next one is the exceptions of Mendelism. Exceptions of Mendelism. First one is the incomplete dominance. Just now in the monohybrid cross we are seeing only the two kind of character. One is a dominant one, another one is a recessive one. Expressing the dominant, not expressing means that is called as a recessive one. So exception means some cases are an exception. That is a very rare case. First one is a incomplete dominance, another one is a co-dominance. Today we will discuss only the incomplete dominance. Co-dominance will be discussed. Tomorrow we will discuss the co-dominance. The last small subtopic is a incomplete dominance. So incomplete means what's the meaning of incomplete? Complete means completely fulfill. That is called as a complete. Incomplete means does not complete. New things will be formed. Does not complete. It does not fulfill. That is called as a incomplete. So dominant is not complete. That is called as a incomplete one. Incomplete one. For example, simply we can say new character will be formed. New character will be formed. That is called as a incomplete dominance. That incomplete dominance are occurs in the various plants, dog, flora, etc., etc. For example, if you can that incomplete dominance we can observe in the 4 o'clock plant. 4 o'clock plant, Miraphilus jalapa. Miraphilus jalapa. That incomplete dominance occurs in the Miraphilus jalapa. That is commonly known as a 4 o'clock plant. Here, what will happen means caps are smaller. Caps are smaller. That is the end of the result. The parental gametes, parental zygotes are caps or caps are. That is a red color flower. Caps are is a red color flower. Small r is a white color flower. Small r is responsible for the production of a white color flower. This is dominant one. The next one is that gamete will be separated like this. Caps are will be separated. Smaller will be separated. The next one is a gamete will be formed. Smaller and smaller. So end of the result, we can get it caps R and small r. Caps R and small r. So caps R and small r, which is a pink color flower. So again I repeat, caps R are responsible for the red color flower production. Small r is responsible for the white color flower. So end of that gametes will be separated. Caps R and small r. That both gametes will be one gamete will be entered into another one and zygote will be formed. So at that time, finally, we can get it a heterozygous gamete. Caps R and small r. Caps R and small r. So caps R and small r is responsible for the pink color flower production. Pink color. So end of the result, we got a pink color flower. So end of the result, what should we get it? Either the parental character should be expressed, either that dominant character red or recessive character white should be exposed. But here, instead of that two character, new character is formed, pink color which is formed. That pink color is exposed. So this is called as a incomplete dominance. That both the characters will be mixed together and new characters will be formed. Pink, red and white will be mingled together and pink color will be formed. That character is exposed in the F1 generation. F1 generation. So this is called as a incomplete. That is called as a incomplete dominance. Clear? Incomplete dominance. That incomplete dominance also undergoes a selfing process like a monohybrid process, which is undergoes a selfing process. Selfing means that same F1 generation result will be again undergoes a self-pollination. Caps R and small r will be undergoes a self-pollination. So finally we can get it a 1:2:1 ratio. So incomplete dominance ratio is a 1:2:1. That is a ratio of incomplete dominance. Phenotypic ratio is a 1:2:1. That is the ratio of phenotypic ratio of incomplete dominance. Here, clear, ma? So incomplete dominance means nothing, new character will be formed. So that's all about today's class, ma. So today, what are the things we discussed means? So first of all, I gave an introduction about the inheritance and variations. Inheritance and variations. So inheritance and variations. And variations, two types of variations I explained. One is a genetic variation, another one is a environmental factor variations. I mean phenotypic variations. The next one is gave definition for genes, heredity, test cross, back cross, or etc. etc. So the important scientist name is Batson and Krieger-Johann-Mentel. That is the two important things. And uniqueness, reasons for Mendel's success, test cross, back cross, monohybrids, and two laws, exception of Mendel's law, incomplete dominance. Those things only we discussed. Clear? Thank you. Tomorrow we will discuss about the remaining topics. Thank you.